Hi everyone, thanks for watching this video presentation. My name is Dave Madge and I'm here to tell you a little about our platform supporting infectious disease drug discovery at WuXi AppTec. I'll kick off with a few words about WuXi AppTec. We're a global drug discovery solutions provider with headquarters in China and 29 sites around the world. I'm based in Cambridge in the UK and look after our collaborators in Europe and Israel. We support diverse organizations in their drug discovery activities, including universities, small biotech companies, and multinational pharma companies. These companies benefit from the comprehensive discovery platforms we've built and the flexible and efficient working practices that they enable. Our philosophy is to build end-to-end solutions to support our collaborators, providing chemistry, biology, DMPK, toxicology, and manufacturing support as their projects move along the development pathway from discovery to clinic. We support all therapeutic areas and diverse therapeutic modalities. This slide provides a high-level overview of the technologies we have assembled, including a wide range of early-stage enabling technologies for target selection and validation, hit finding and computational support, then moving into the drug discovery phase with support from medicinal chemistry, in vitro biology, in vitro ADME, and as projects progress, in vivo pharmacokinetics and pharmacodynamics. To support these activities, we have very comprehensive discovery, chemistry, and biology capabilities. And I'll talk about these in the following slides, with a focus on our capabilities in infectious disease drug discovery. There are often some key building blocks that really enable discovery projects, and these might include access to genetic manipulation tools to create cell lines and validate targets, or to support assays, as well as synthesis and purification of purified proteins for activity studies and for structural biology. We have built a comprehensive platform supporting these activities, generating thousands of cell lines and many hundreds of proteins across all target classes. Many of these materials will find their way into one or more of the tools we've built to help identify new chemical matter to form the starting point for drug discovery programs. These include affinity-based selection platforms such as ASMS and DNA-encoded libraries, as well as fragment screens and functional assays with biochemical or phenotypic endpoints. We're also increasingly using virtual screening techniques, especially for targets with pre-existing structural ligand information, and we're able to screen billions of virtual compounds to identify a subset for rapid synthesis and testing as an alternative hit-finding strategy. To back up these hit-finding tools, we've built the world's largest and most experienced chemistry organization, enabling fast and efficient synthesis of a wide range of molecule types on scales supporting early-stage discovery, as well as later-stage in vivo pharmacology and toxicology studies. Last year, our teams generated 24 preclinical candidates on behalf of our clients and supported lead optimization on hundreds of programs. To test these molecules, we've built in vitro biology capabilities that support all target classes with diverse biophysical, biochemical, and functional readouts, generating millions of data points every year in efficacy, mechanism of action, and SAR development studies. Turning now to the ways in which we can support drug discovery for infectious disease targets, this slide provides an overview of the scope of capabilities from development of supporting tools such as cell lines and assays, high throughput screening to identify new chemical matter, support to lead optimization programs with SAR development and mechanism of action studies, as well as support to clinical development programs with clinical virology, genotyping, and sequencing. We supported most target classes and do so with the creation of bespoke packages to meet diverse client requirements. I'm going to give some examples across three main target classes, being hepatitis B, influenza, and COVID-19 assays. To start with hepatitis B, we've been supporting cell-based assays, in vivo studies, and clinical virology. For drug screening, we have a range of HPV-producing cell lines and can analyze with reporter assays, protein quantification, and PCR, for example, looking at core proteins and capsid assembly. We have animal models across multiple species, including transgenic and humanized mice, and support clinical virology with genome sequencing, phenotyping, cytokine analysis, and antibody assays. I'll show some reference data for some of these assays over the next few slides. Firstly, here is some data collected from primary human hepatocytes with the antivirals lamivudine and entecavir, showing robust reduction in HBV DNA copy count, with supporting IHC images demonstrating that we can not only look at the endpoint, but also look at tissue effects. We show some further data on this slide using FRG humanized mouse model with immunohistochemistry staining to show the effect of entecavir on plasma HBV 3 weeks after HBV infection and during a 2-week treatment with this antiviral agent. You can see a dose-dependent reduction in terminal HBV levels in the liver in the center graphic, supported by plasma data on the left and IHC data on the right. We also have the ELISpot assay platform, which we've been using to look at activation of HBV-specific T cells in the mouse model. This model uses ex vivo stimulation of lymphocytes and looks, for example, at the production of cytokines such as interferon gamma, with challenges with HBV-derived proteins or peptides. We can also track antibody production in this assay, which is illustrated on this slide for the anti-hepatitis B antibodies. Moving on to influenza studies, again, a range of in vitro and in vivo support with screening and compound profiling assays, biochemistry, animal models, and in particular models in the mouse and the ferret. To give a few examples of these, in the mouse model, we're supporting H1N1, H3N2, and Leaf40 assays with a range of dosing routes for small molecules and other modalities, and readouts including body weight, viral load, serum antibodies, and immunohistochemistry. Oseltamivir and baloxavir are often the reference substances for these assays, and I show here some data for these molecules in the H1N1 model with survival and body weight data. In the lower panel, some data with an experimental flu vaccine showing the protection in the far right panel provided by a high-dose vaccine in this model. We can also build cytokine profiling into these assays, and I show here representative data collected with oseltamivir, looking at impacts on IL-6 and TNF-alpha in an influenza mouse model. In this last section of the presentation, I'd like to discuss assays we've developed and are using to support efforts to identify treatments for the COVID-19 virus. These assays fall into 2 main groups, those directed towards the general human coronavirus and those directed specifically towards SARS-CoV-2. We have both cell-based and enzymatic assays, and I'll provide a few examples on the next few slides. For HCOV-229E, we're using RT-qPCR and cytopathic effect, or CPE, assays in MRC5 cells. On this slide, you can see the effect of references such as remdesivir, chloroquine, and ribavirin in these cells after 2 days in the RT-PCR assay or 3 days in the CPE assay. These are high-throughput plate-based assays, so we can deal with many hundreds of compounds in these assays. We also run these assays in the hepatocyte carcinoma HUH7 cells, typically over 4 days with cell viability as the endpoint. For HCOV-OC43, we validated assays also in HUH7 cells with CPE and PCR as the endpoints and with remdesivir and chloroquine as the references. Turning to some of the specific enzyme assays recently developed, the main protease, or MPRO, is assayed with a plate-based freight assay. This main protease assay is available for several related pathogens, including SARS, MERS, and NL63. Assays have also been developed for the papain-like protease PLPRO in microtiter plate format, as well as an assay for the RNA-dependent RNA polymerase, which can be assayed with a simple enzyme assay with ATP as the substrate, as shown on this slide. Finally, we've also developed both the spike pseudovirus assay and immunofluorescence assays, IFA assays, for SARS-CoV-2. I'll just briefly conclude by saying that supporting the chemistry and biology platforms I've described here, we also support all our drug discovery programs with a comprehensive suite of metabolism and pharmacokinetic assays, including IND-enabling studies. As well as this, we have a full toxicology capability, providing IND-enabling tox studies across all species, genotoxicity, cardiotoxicity, and tolerance studies. We're also a CAP-accredited molecular testing laboratory, providing molecular testing support to clinical studies, including genomics, clinical virology, studies with oncolytic virus, vaccines, modified T cells, and stem cell therapies. So thank you very much for your attention today. Please feel free to contact me with questions or inquiries, and I very much appreciate your attention. Bye-bye.